Hello everyone, and welcome to this lesson on photosynthesis and the chloroplast. As we know, living organisms don't live passively. They are quite active. They need to carry out various different metabolic activities, and these activities will use up energy. This energy is going to be in the form of ATP. But now the question is, where does that energy originally come from? As you may know from physics, energy can't be created or destroyed, but can only be changed from one form to another. So all the energy that exists in living organisms needs to come from somewhere. And that somewhere is going to be the sun. You see, the sun is the primary source of energy for all living organisms. And what happens is that the light energy from the sun gets converted into chemical energy by the process of photosynthesis, and this happens in green plants. You see, some organisms, which are mainly going to be your green plants, but also include certain types of algae and certain single-celled organisms, have this ability, where they can convert an inorganic form of carbon, such as carbon dioxide, into an organic form of carbon. And they do this using the sun's energy. So the light energy from the sun gets converted into the chemical energy in the bonds of the organic form of carbon. And this is going to be what we call the process of photosynthesis. You are synthesizing this organic form of carbon, such as a sugar, for example, using light, right? Photo is light and synthesis. Now, as we mentioned, the main organisms that are going to be carrying out photosynthesis are going to be your green plants. But have you ever wondered why these plants are green in the first place? The reason for that is mainly because they have in the chloroplasts of their cells a certain pigment called chlorophyll, and that is responsible for giving them this green color. It's the job of this chlorophyll, this pigment, to absorb the light energy that's coming from the sun. And as we mentioned, this chlorophyll is found inside the chloroplasts. These chloroplasts, if you remember the endosymbiont theory, were one of the two organelles, along with the mitochondria, that were thought to once have been independent prokaryotic cells, that eventually, over time, started to live inside the eukaryotic cells. This explains why the chloroplasts have a structure that is very similar to that of a prokaryotic cell. So, for example, they have two membranes that make up an envelope, right? Inside they have DNA. They have something like a cytoplasm, which is called the stroma. They have ribosomes, and they have some starch grains. It's a very similar structure to that of a prokaryotic cell. Now, when you look at a plant cell under a light microscope, you can see the chloroplasts, and they appear as a green biconcave structure. However, not much detail beyond that is visible. To see a detailed structure of a chloroplast, we need to use an electron microscope. When we do this, we clearly see these little biconcave structures that are about 4 to 10 micrometers in length and about 2 to 3 micrometers wide. Since they once used to be prokaryotes, we can see that they are surrounded by a membrane, and if you look closely, you'll see that it's actually two membranes that form a chloroplast envelope. Then, inside those membranes, you have this ground substance that is kind of like the cytoplasm of the chloroplast, which is called the stroma. And in this stroma is where you find the various structures like, for example, the DNA, which is going to be naked circular DNA. You find some ribosomes, which are going to be 70S ribosomes. You find the various enzymes that are going to be required for photosynthesis. You find certain things like starch grains and lipid droplets, and you find what are called grana and thylakoid membranes, and we'll talk about what those are in a minute. Before that though, let's do a quick overview first about what photosynthesis is and how it works. At a glance, if you just look at this equation for photosynthesis, you can see that what's happening here is that you're taking carbon dioxide and water and using the energy that you get from sunlight, which was absorbed by chlorophyll, remember, to convert this stuff into glucose and producing oxygen as a waste product. This, if you notice, at least by looking at the equation, seems to be the complete opposite of what we're doing with respiration. In the case of respiration, you take glucose and you use oxygen to oxidize it to produce energy in the form of ATP, as well as some carbon dioxide and water as waste products. So that's the complete opposite of what you're doing in photosynthesis. That being said, keep in mind though that these equations are just a basic summary of the entire process, right? Kind of like a balance sheet of what goes in and what you get out the other end. This is not the entire process. The entire process actually consists of multiple different stages, and each of those stages has multiple different steps within it, and we'll cover all of that in separate videos. For now though, in this lesson, I just want to introduce you to the basic idea that the entire process of photosynthesis can be divided into two sets of reactions. One of these is going to be what are called the light-dependent reactions, and the other are going to be the light-independent reactions. And both of these sets of reactions happen in the chloroplast. The light-dependent reactions, as the name suggests, are dependent on light, meaning they need light to proceed. These reactions are going to involve several membrane-bound proteins, some of which are going to behave as pigments, some of which are going to make up an electron transport chain, and some of which are going to make up ATP synthases. These, of course, will be used to produce ATP. Now, in this reaction, one of the important things that's going to happen is that the energy from light is going to be used to split water molecules in order to produce hydrogen ions, electrons, and oxygen. This process where you split a water molecule using light is called photolysis. Photo for light and lysis for breaking something down. So you're breaking down the water molecule using light. So it's called photolysis. From this process, you've now formed hydrogen ions, electrons, and oxygen. Now, the oxygen is going to be a waste product, so this is not going to be used anymore for photosynthesis. This we're done with. What we have left now is the hydrogen ions and the electrons from the photolysis of water, and if you remember, that ATP that was generated from the ATP synthase. These are some of the products of the light-dependent reactions. Now, the ATP is going to move forward and be used now in the light-independent reactions. However, the electrons and hydrogen ions are instead going to be accepted by a carrier molecule called NADP. This molecule, when it accepts the hydrogen ions and the electrons, becomes reduced. And this reduced form of NADP can also be written as NADPH. And it's this NADPH now that's going to move from the light-dependent reactions to now be used in the light-independent reactions along with the ATP. So the big picture of what's happening here is that the ATP and the NADPH that is produced from the light-dependent reactions is moving forward now to be used in the light-independent reactions. Okay, but now another question is, where exactly in the chloroplast are these light-dependent reactions even taking place? If you remember, earlier we said that the chloroplast has a double membrane that makes up its envelope. There's an outer membrane and there's an inner membrane. And between the two membranes is what is called the intermembrane space. Then, within the inner membrane, you have this ground substance known as the stroma. It's actually within this stroma that you have another system of membranes. This system consists of a series of flattened fluid-filled sacs called thylakoids. And in certain places, these thylakoids form stacks, right? And a stack of these thylakoids is called a granum, the plural of which is going to be grana. These grana are then connected to each other by these tubes of membranes called lamellae, the singular of which will be lamella. So again, the fluid-filled sacs are called thylakoids, the stacks of thylakoids are called grana, and the grana are connected to each other by lamellae. And the membranes of all of these structures together are collectively called the thylakoid membranes. It's on these thylakoid membranes that you'll find the membrane-bound proteins that are required for the light-dependent reactions, because this is where those reactions take place. on the thylakoid membranes.
Quick pause. If you're finding value in this explanation, there's so much more to discover at Alt Academy, where we break down every A-level concept with the same clarity. Now back to the video lesson.
The reason this system of membranes is arranged the way it is, with stacks of thylakoids forming grana and then being connected by lamellae, is because this arrangement helps to increase the surface area to volume ratio of the thylakoid membranes, making these light-dependent reactions more efficient. You see, it's in these thylakoid membranes that you'll find these proteins called photosystems, and these are the proteins that contain the pigments, such as chlorophyll for example, that are responsible for absorbing the light. So the more surface area you have for the thylakoid membranes, the more of these photosystems you can accommodate. And the more of these you have, the more light you can absorb, and therefore the faster the light-dependent reactions can occur. We'll talk in detail about the role of the photosystems and the electron transport chain and so on when we talk about the light-dependent reactions in a separate lesson. For now, just keep in mind that this is going to be taking place on these thylakoid membranes. Now, after the light-dependent reactions, the next part of photosynthesis is going to be the light-independent reactions, and these are going to take place in the stroma of the chloroplast. This set of reactions, as the name suggests, are not directly dependent upon light, meaning light isn't directly involved in these reactions. That being said, the light-independent reactions do still need some of the products that were made from the light-dependent reactions. As we mentioned earlier, the ATP and the NADPH from the light-dependent reactions are going to move forward and be used in these light-independent reactions. It's for this reason that even though this set of reactions doesn't directly need light, it still can't happen in the nighttime, for example, because it needs the products from the light-dependent reactions, which will only be being made during the day when there's light available. Now, in these light-independent reactions, what's essentially happening is that carbon dioxide molecules are going to be used to make glucose, right? Carbohydrates. And of course, to do this, you're going to need to form some carbon-carbon bonds, and that is going to need energy. That's why we need the ATP from the light-dependent reactions to help do this. As these ATPs get used up in order to provide energy, they get converted into ADPs and inorganic phosphates, and those molecules can then go back to the light-dependent reaction to again be used for further ATP synthesis. Okay, so that's why we need the ATP. Now, the other product from the light-dependent reactions that was going to be used in the light-independent reactions was NADPH. Why do you think we need this? Well, if you remember the structure of glucose, it also has hydrogens, right? Where do you think those hydrogens are coming from? You get the carbon from the CO2s, and you get the oxygen from the CO2s, but you don't get any hydrogen from here. Well, you get the hydrogens from the NADPH molecules. If you remember, those molecules accepted some hydrogens earlier during the light-dependent reactions, and now they're going to donate those hydrogens here to help us make carbohydrates, such as glucose. As these NADPH molecules donate their hydrogens here, they again become NADP. So they get oxidized back into NADP. And now they can go back to the light-dependent reactions to accept more hydrogens from there. So, as we've seen, not only are products from the light-dependent reactions, such as ATP and NADPH, going to be used for the light-independent reactions, but products from the light-independent reactions, such as NADP and ADP and inorganic phosphate, are also going to be used for the light-dependent reactions. Since the light-dependent reactions are going to be taking place in the thylakoid membranes, particularly of the grana, and the light-independent reactions are going to be taking place in the stroma of the chloroplast, that means that these substances need to be transported across the thylakoid membrane. So, NADPH and ATP from inside the thylakoid spaces are going to need to be moved across the membrane to enter the stroma, and likewise NADP, ADP, and inorganic phosphate are going to need to move across the membrane to enter into the thylakoid. To facilitate all of this movement so that it can happen quickly and easily, The thylakoid membranes have transporters that can allow all of these substances to easily move across. Now, once you've successfully made glucose from the light-independent reactions of photosynthesis, this glucose can then be combined with fructose to make sucrose. This is going to be the main form of sugar that's going to be transported across the plant through the phloem, the sucrose. Now, alternatively, if the plant wants to store the glucose long term, it can also be converted into starch. This will happen in the stroma of the chloroplasts. This starch, if you look at a plant cell underneath a microscope, and this works for both the light microscope and an electron microscope, can actually be seen. As you can see here, this little starch grain is going to be visible under a microscope. Now, an important property of this stroma, which is where the light-independent reactions, remember, are taking place, is that it is actually a polar substance, which makes it an ideal environment to dissolve all of the enzymes that are needed for the various reactions that will take place here, including the light-independent reactions that we just talked about. Besides these enzymes and some starch grains, in this stroma you can also find some lipid droplets, and these are usually formed from the breakdown of the thylakoid membranes. Then, the stroma is also going to contain the chloroplast's DNA, which is going to be naked circular DNA. This DNA, along with the 70S ribosomes that are also present in the stroma, mean that the chloroplast is able to make its own proteins, such as the enzymes that we find here, and divide on its own, independently of the rest of the plant cell. These abilities of the chloroplast are important because this means that in those parts of the plant where you need more photosynthesis to occur, the chloroplasts can divide to increase their numbers and actually provide that level of photosynthesis. So, in the parts of the plant where a high level of photosynthesis needs to take place, such as, for example, the palisade mesophyll cells, the chloroplasts will divide and increase their number so that they can provide that level of photosynthesis. So, as we've seen, the structure of these chloroplasts is very closely associated to their function, and that function is very important. So these are pretty important organelles. Alright, as a quick recap, in this lesson we said that the main source of energy for living organisms is ultimately going to be the sun, and that light energy is going to be captured using the process of photosynthesis. In this process, the light energy from the sun is going to be used to convert inorganic carbon into an organic form of carbon. So the sun's light energy is going to become the chemical energy in the bonds of that organic molecule. This light energy is going to be absorbed by a pigment in plants called chlorophyll, which is responsible for giving the plants their green color. This chlorophyll is going to be found inside chloroplasts, which are little green biconcave organelles about 4 to 10 micrometers long and 2 to 3 micrometers wide. They have two membranes that make up their chloroplast envelope, and within the inner membrane you have some ground substance known as the stroma. It's within this stroma that you'll find the naked circular DNA, the 70S ribosomes, the enzymes that are needed for photosynthesis, especially the light-independent stage. You'll find starch grains, lipid droplets, and you'll find thylakoid membranes, including the grana. When we look at the equation for photosynthesis, we can see that what's happening here is that light energy is being used to convert carbon dioxide and water into glucose, producing oxygen as a waste product. This entire process can be divided into two sets of reactions, the light-dependent reactions and the light-independent reactions, and both of these groups of reactions take place in the chloroplast. The light-dependent reactions need light, and they involve pigments, the electron transport chain, and the enzyme ATP synthase. In this process, light energy will be used to split water molecules into hydrogen ions, electrons, and oxygen in a process called photolysis, while ATP will be synthesized using the enzyme ATP synthase. The hydrogen ions and electrons will be accepted by a carrier molecule called NADP to form NADPH, and then this NADPH and ATP will both be moving forward towards the light-independent stages to be used there. Now, the light-dependent reactions take place on a membrane system that is found within the stroma of the chloroplasts called the thylakoid membranes. This system consists of a series of flattened fluid-filled sacs called thylakoids. In places, these thylakoids form stacks called grana, and the grana can be joined to one another by these membrane tubes called lamellae. This arrangement increases the surface area to volume ratio of the thylakoid membranes, making the light-dependent reactions more efficient, because it's on these membranes that all those membrane-bound proteins that are needed for the light-dependent stage are going to be found. The next stage, the light-independent reactions of photosynthesis, are going to take place in the stroma of the chloroplast. In this stage, ATP and hydrogens donated from NADPH are going to be used to convert carbon dioxide into glucose. This will reform NADP as well as ADP and inorganic phosphate, which can then go back and play their role again in the light-dependent stages. Once you've formed glucose from the light-independent reactions, this glucose can then either be combined with fructose to form sucrose, which can then be transported in the phloem, or it can be converted into starch to be stored long-term. This starch can even be seen under a microscope as these little starch grains. Besides these starch grains, the polar stroma inside the chloroplasts also contains all the dissolved enzymes that are going to be needed for all the reactions of the light-independent stage. Besides this, it also contains the DNA and 70S ribosomes, which give the chloroplasts the ability to make their own proteins and to divide independently. This ability means that in the parts of the plant where you have a higher demand for photosynthesis, such as the palisade mesophyll cells, the chloroplasts can divide and accommodate so that they can actually provide that level of photosynthesis. We'll go into detail about all the different stages of photosynthesis in separate lessons. But for now, this lesson has come to an end. I hope you found it useful, and I'll see you in the next one.
Hey there! If you found this A-level biology lesson helpful, why don't you head over to altacademy.org and sign up for our full A-level biology course. In addition to biology, you could sign up for physics, chemistry, math, or 4 other A-level subjects. Thousands of students have already taken the leap. What are you waiting for? See you there.